Notes. A. Lymphocyte trafficking. Answer. Lymphocyte trafficking is the continuous circulation of lymphocytes between the bloodstream and secondary lymphoid organs. This process is essential for immune surveillance, allowing T and B cells to scan for specific antigens. B. Inflammation. Answer. Inflammation is a protective biological response of vascular tissues to harmful stimuli, such as pathogens, damaged cells, or irritants. It involves immune cells, blood vessels, and molecular mediators and typically presents as redness, swelling, heat, pain, and loss of function. C. Haptens. Answer. Haptens are small molecules that can bind to specific antibodies but cannot induce an immune response on their own. They become immunogenic only when coupled to a larger carrier molecule, such as a protein. D. Memory cells. Answer. Memory cells are long-lived B and T lymphocytes generated after an initial immune response. They enable a faster and stronger secondary immune response upon subsequent exposure to the same antigen. E. ELISA. Answer. ELISA, enzyme-linked immunosorbent assay, is a laboratory test that uses antibodies in color change to identify a substance, typically an antigen or an antibody, in a sample. It is widely used in medical diagnostics and research. 3. Describe the mechanism of T cell activation and differentiation. T cell activation and differentiation. Step 1. Antigen presentation. T cell activation begins when an antigen-presenting cell, APC, such as a dendritic cell, internalizes and processes an antigen into peptides. These peptides are then presented on the APC's surface within major histocompatibility complex, MHC, molecules. Step 2. T cell receptor, TCR, binding and co-stimulation. A specific T cell receptor, TCR, on the T cell surface binds to the MHC peptide complex, signal 1. Simultaneously, co-stimulatory molecules, such as B7 on the APC and CD28 on the T cell, interact, signal 2. Both signals are required for full activation. Step 3. Cytokine signaling. The interaction of signals 1 and 2 triggers intracellular signaling pathways, leading to the production of cytokines, most notably interleukin-2, IL-2. IL-2 acts in an autocrine manner to promote T cell proliferation and differentiation. Step 4. Clonal expansion and differentiation. The activated T cells undergo rapid clonal expansion and differentiate into various effector and memory T cell subsets, such as helper T cells, MathBF, CD4+, and cytotoxic T cells, MathBF, CD8+, each with specialized functions. Answer. The mechanism involves antigen presentation by APCs via MHC molecules, followed by TCR binding, signal 1, and co-stimulatory signals, signal 2, which together induce cytokine production, like IL-2, and subsequent T cell proliferation and differentiation into effector and memory cells. 4. What do you mean by antigenicity and immunogenicity? Antigenicity and immunogenicity. Step 1. Define antigenicity. Antigenicity is the ability of a substance, antigen, to bind specifically to immune components like antibodies or T cell receptors. Step 2. Define immunogenicity. Immunogenicity is the ability of a substance to induce a humoral and/or cell-mediated immune response. Step 3. Highlight the relationship. All immunogens are antigens, but not all antigens are immunogens. A molecule must be large and complex enough to be immunogenic, while smaller haptens are antigenic but not immunogenic on their own. Answer. Antigenicity is the capacity to bind to immune effectors, while immunogenicity is the capacity to induce an immune response. All immunogens are, MathBF, antigenic, but not all antigens are, MathBF, immunogenic. 5. Describe the factors influencing antigenicity and immunogenicity. Factors influencing antigenicity and immunogenicity. Step 1. Foreignness. The immune system distinguishes between self and non-self. The greater the phylogenetic difference from the host, the more foreign and thus more immunogenic a substance is likely to be. Step 2. Molecular size. Generally, larger molecules are better immunogens. Molecules with a molecular weight of less than 10,000 Daltons are typically poor immunogens. Step 3. Chemical complexity and composition. Complex molecules with diverse chemical structures, e.g., proteins with multiple amino acids, are more immunogenic than simple homopolymers. Step 4. Degradability and physical form. Antigens must be degradable by APCs to be presented on MHC molecules. Particulate antigens are generally more immunogenic than soluble ones. Step 5. Genetic factors and dosage. Host genes, MHC molecules, and the dose, route, and timing of antigen administration also significantly influence the immune response. Answer. Factors include foreignness, molecular size, chemical complexity, degradability, physical form, host genetic factors, and the dose and route of administration. 6. Give an account of the mechanism of activation of complement system. Activation of the complement system. Step 1. Overview of pathways. The complement system can be activated through three main pathways: the classical, the alternative, and the lectin pathways. All three converge at the activation of component C3. Step 2. Classical pathway. Triggered by antibody-antigen complexes binding C1, leading to the formation of MATBF, C3, convertase, C4b2A. Step 3. Lectin pathway. Initiated by mannose binding lectin, MBL, binding to microbial carbohydrates, also forming the MATBF, C3, convertase, C4b2A. Step 4. Alternative pathway. Activated spontaneously by the hydrolysis of C3, which binds to microbial surfaces and forms the MATBF, C3, convertase, C3BBB. Step 5. Common terminal pathway. All C3 converters cleave C3 into C3A and C3B. C3b then initiates the formation of C5 convertase, which leads to the assembly of the membrane attack complex, MAC, resulting in cell lysis. Answer. The complement system activates via the classical, antibody-mediated, lectin, MBL-mediated, and alternative, spontaneous, pathways, all converging to form MATBF, C3, convertase, which leads to the formation of the membrane attack complex, MAC, and target cell lysis. 7. What is the genetic basis of generation of antibody diversity? Explain. Genetic basis of antibody diversity. Step 1. V-D-J recombination. The primary mechanism is V-D-J recombination, where variable V, diversity, D, and joining, J, gene segments are randomly rearranged and joined by the RAG enzymes to form a unique functional gene for the antibody variable region. Step 2. Junctional diversity. The process of V-D-J recombination is imprecise, with the random addition or subtraction of nucleotides at the junctions between gene segments by the enzyme TDT. This creates significant junctional diversity. Step 3. Somatic hypermutation. After antigen exposure, B cells undergo somatic hypermutation in germinal centers, introducing point mutations into the V regions of the antibody genes, which increases antibody affinity. Step 4. Class switch recombination. Antibodies can change their constant region, e.g., from IgM to IgG, through class switch recombination, altering their effector function without changing antigen specificity. Answer. The genetic basis of antibody diversity is primarily V-D-J recombination, which is enhanced by junctional diversity, somatic hypermutation, and class switch recombination. 8. Give an account of immune responses generated upon viral infections. Immune responses to viral infections. Step 1. Innate immunity, interferons and NK cells. The initial response involves the production of type 1 interferons, which induce an antiviral state in surrounding cells, and natural killer, NK, cells, which can directly kill infected cells. Step 2. Humoral immunity, antibodies. B cells produce neutralizing antibodies that block viruses from entering host cells and target free virus particles for destruction. Step 3. cell-mediated immunity, cytotoxic T cells. Cytotoxic T lymphocytes, MATBF, CD8+, recognize and kill host cells that have been infected with the virus by detecting viral antigens presented on the cell surface via MHC class I molecules. Step 4. Memory response. The infection generates long-lived memory T and B cells, which provide protection against future re-exposure to the same virus. Answer. Immune responses to viral infections involve immediate innate immunity, interferons, NK cells, followed by adaptive immunity, including neutralizing antibodies, humoral, and cytotoxic T lymphocytes, MATBF, CD8+, that kill infected cells. AI responses may include mistakes. Answers might be inaccurate or misleading. Double check for accuracy. Two. Short notes. A. Ovum transport. Answer. Ovum transport refers to the journey of the egg from the ovary through the fallopian tube to the uterus. 1. After ovulation, the ovum is released from the ovary and captured by the fimbriae of the fallopian tube. 2. Cilia lining the fallopian tube in muscular contractions, peristalsis, move the ovum towards the uterus. 3. Fertilization typically occurs in the ampulla region of the fallopian tube. And 4. The entire process usually takes about 3 to 4 days. B. Immunocontraception. Answer. Immunocontraception is a method of birth control that uses the body's immune response to prevent fertilization or implantation. 1. It often involves vaccines that target specific reproductive hormones, like GnRH or HCG, or sperm, egg surface proteins. 2. The immune system produces antibodies that interfere with normal reproductive processes. And 3. This method is primary. 2b. Immunocontraception. Answer. Immunocontraception is a method of birth control that uses the body's immune system to prevent conception. This approach typically involves vaccines that target specific reproductive hormones, like HCG, or proteins on the surface of sperm or eggs, triggering an immune response that interferes with fertilization or implantation. 2c. Types of stem cells. Answer. Stem cells are categorized based on their potency, ability to differentiate into different cell types. The main types include: 1. Totipotent, can differentiate into all cell types, including the placenta, e.g., zygote. 2. Pluripotent, can differentiate into all cell types of the 3 germ layers, but not the placenta, e.g., embryonic stem cells. 3. Multipotent, can develop into a limited number of cell types within a specific lineage, e.g., hematopoietic stem cells. 4. Oligopotent, can differentiate into a few cell types, e.g., lymphoid or myeloid stem cells. And 5. Unipotent, can only produce one cell type, e.g., muscle stem cells. 2d. ICSC. Answer. ICSC stands for intracytoplasmic sperm injection. It is an assisted reproductive technology, ART, procedure in which a single sperm is directly injected into the cytoplasm of an egg to achieve fertilization. This method is often used to overcome male infertility issues. 2e. Therapeutic cloning. Answer. Therapeutic cloning is the process of creating cloned embryos for the purpose of extracting stem cells. The goal is to generate embryonic stem cells that are genetically identical to the patient, which can then be used to treat diseases or repair damaged tissues without the risk of immune rejection. 3. Process of ovarian follicle growth and differentiation. Step 1. Primordial follicle activation. The process begins with the activation of primordial follicles into primary follicles, a process independent of gonadotropins. Step 2. Follicle growth and differentiation. The primary follicle develops into a secondary follicle, characterized by the formation of the zona pellucida and the accumulation of follicular fluid in a space called the antrum. Step 3. Hormonal influence and maturation. Under the influence of follicle-stimulating hormone, FSH, and luteinizing hormone, LH, the secondary follicle matures into a tertiary, Graafian, follicle. The oocyte inside completes meiosis I and arrests in metaphase 2. Step 4. Ovulation and corpus luteum formation. An LH surge triggers ovulation, releasing the secondary oocyte. The remaining follicular cells transform into the corpus luteum, which produces hormones like progesterone. Answer. The process of ovarian follicle growth and differentiation involves the transition from primordial to primary, secondary, and finally tertiary, Graafian, follicles under hormonal control, culminating in ovulation and the formation of the corpus luteum. 4. Process of oogenesis. Step 1. Oogonia proliferation. Oogenesis starts in the fetal ovary with the proliferation of oogonia, diploid stem cells, through mitosis. Step 2. Primary oocyte formation and meiosis I arrest. Oogonia develop into primary oocytes, which initiate meiosis I but arrest at prophase I before birth. Step 3. Meiosis I completion. At puberty, during each menstrual cycle, a few primary oocytes complete meiosis I, forming a large secondary oocyte and a small first polar body. Step 4. Meiosis II arrest and completion. The secondary oocyte begins meiosis II but arrests at metaphase 2. Meiosis II is only completed upon fertilization by a sperm, resulting in a fertilized ovum, zygote, and a second polar body. Answer. Oogenesis is the process of female gamete formation, involving mitosis of oogonia, followed by two meiotic divisions that produce a primary oocyte, a secondary oocyte, and polar bodies, ultimately yielding a single functional ovum upon fertilization. 5. Role of cytoplasm and nucleus in cellular differentiation. Answer. The nucleus contains the genetic material, DNA, that dictates all potential cell fates, while the cytoplasm contains regulatory molecules that control gene expression, thus determining the specific path of differentiation. The nucleus provides the blueprint for all cell types. The cytoplasm, however, contains maternal mRNAs and proteins that are unequally distributed during cell division. These cytoplasmic factors act on nuclear genes, turning specific ones on or off and steering the cell towards a specialized function. 6. Process of limb generation in vertebrates. Answer. Limb generation, regeneration, in vertebrates is a complex process that involves dedifferentiation of existing cells to form a blastema, followed by coordinated cell proliferation and redifferentiation. The process typically involves 1. Wound healing, rapid closure of the wound epithelium. 2. Dedifferentiation, formation of a mass of progenitor cells called the blastema. 3. Pattern formation. The blastema cells proliferate and differentiate into new bone, muscle, and other tissues based on positional information. And 4. Growth and morphogenesis. The new structures grow and integrate with the existing body plan. 7. Nuclear reprogramming of pluripotent stem cells. Step 1. Identification of key transcription factors. The process, famously pioneered by Yamanaka, involves identifying a specific set of transcription factors, commonly OCT4, SOX2, KLF4, and CMYC, that can induce pluripotency. Step 2. Delivery of factors. These factors are introduced into differentiated somatic cells, like skin cells, using viral vectors or other delivery methods. Step 3. Induction of reprogramming. The introduced factors alter the cell's gene expression profile, gradually silencing differentiation-associated genes and activating pluripotency genes. Step 4. Selection and characterization. The reprogrammed cells, called induced pluripotent stem cells, iPSCs, are selected and characterized to confirm their pluripotent state and ability to differentiate into various cell types. Answer. Nuclear reprogramming of pluripotent stem cells is achieved by introducing specific transcription factors, Yamanaka factors, into somatic cells, which induces a change in gene expression, leading to the formation of induced pluripotent stem cells, iPSCs, that can differentiate into any cell type. AI responses may include mistakes. Answers might be inaccurate or misleading. Double check for accuracy. 2. Short notes. A. Pre and prohormone. Answer. Preprohormones and prohormones. Preprohormones are the initial, inactive polypeptide chains synthesized on ribosomes, containing a signal sequence that directs them into the endoplasmic reticulum. Once inside, the signal sequence is cleaved, converting the preprohormone into a prohormone. Prohormones are also biologically inactive and are then packaged into vesicles in the Golgi apparatus, where they are cleaved by specific enzymes into their active hormone form and other peptide fragments just before secretion. B. Supraoptic and paraventricular nuclei. Answer. Supraoptic and paraventricular nuclei. These are clusters of neurosecretory cells located in the hypothalamus of the brain. The supraoptic nuclei primarily produce the hormone antidiuretic hormone, ADH, while the paraventricular nuclei mainly produce oxytocin. These hormones are then transported down the axons of these neurons to the posterior pituitary gland, where they are stored and released into the bloodstream. C. Beta-adrenergic receptor. Answer. Beta-adrenergic receptor. Beta-adrenergic receptors, beta receptors, are a class of G-protein coupled receptors that bind to the neurotransmitters norepinephrine and epinephrine, adrenaline. There are three main subtypes, beta-1, beta-2, beta-3, each mediating different physiological responses. For example, beta-1 receptors in the heart increase heart rate and contraction force, while beta-2 receptors in the lungs cause bronchodilation. D. Calmodulin. Answer. Calmodulin. Calmodulin, CAM, is a ubiquitous calcium-binding protein found in eukaryotic cells. It acts as an intracellular calcium receptor and plays a crucial role in signal transduction. When calcium ion levels rise in the cell, calcium binds to calmodulin, changing its conformation. This activated CAM then interacts with and regulates the activity of various target proteins, such as protein kinases and phosphatases, to modulate cellular processes like muscle contraction, neurotransmission, and inflammation. E. G protein. Answer. G protein. G proteins, guanine nucleotide binding proteins, are molecular switches involved in transmitting signals from outside the cell to its interior. They are coupled to G protein coupled receptors, GPCRs, in the cell membrane. When a hormone or ligand binds to a GPCR, it activates the G protein, causing it to exchange GDP for GTP. The activated G protein then dissociates into subunits that interact with other enzymes or ion channels to initiate a cellular response. 3. Define neurosecretion. Discuss the role of neurosecretion in the regulation of endocrine system. Answer. Neurosecretion. Neurosecretion is the process by which specialized nerve cells, called neurosecretory cells, synthesize and release hormones directly into the bloodstream. Unlike typical neurons that release neurotransmitters into synapses, these cells translate neural signals into hormonal messages. 1. Hypothalamus pituitary axis. Neurosecretion is central to endocrine regulation, primarily through the hypothalamus pituitary axis. 2. Hypothalamic hormones. The hypothalamus secretes releasing and inhibiting hormones, e.g., GnRH, CRH, TRH, into the portal system, controlling hormone release from the anterior pituitary gland. 3. Posterior pituitary hormones. Neurosecretory cells in the supraoptic and paraventricular nuclei produce ADH and oxytocin, which are stored in and released from the posterior pituitary gland. And 4. Integration of systems. This mechanism effectively integrates the nervous system with the endocrine system, allowing neural input to directly modulate hormonal output and maintain homeostasis. 4. Give an account of hormonal regulation of menstrual cycle. Answer. Hormonal regulation of menstrual cycle. The menstrual cycle is regulated by a complex interplay of hormones from the hypothalamus, pituitary gland, and ovaries. 1. GnRH. The hypothalamus secretes gonadotropin-releasing hormone, GnRH, which stimulates the anterior pituitary. 2. FSH and LH. The pituitary releases follicle-stimulating hormone, FSH, and luteinizing hormone, LH, which act on the ovaries to promote follicle development and ovulation. And 3. Estrogen and progesterone. Ovarian follicles produce estrogen, and after ovulation, the corpus luteum produces progesterone. These hormones regulate the uterine lining in feedback to the hypothalamus and pituitary to control the cycle's progression. 5. Detail the steps involved in the biosynthesis of steroid hormone. Answer. Biosynthesis of steroid hormone. Steroid hormones are synthesized from cholesterol through a series of enzymatic steps, primarily within the mitochondria and smooth endoplasmic reticulum of endocrine cells. 1. Cholesterol uptake, synthesis. Cells obtain cholesterol from the bloodstream, LDL, or synthesize it de novo. 2. Rate limiting step. Cholesterol is transported into the inner mitochondrial membrane, where the enzyme cholesterol desmolase, P450 SCC, converts it to pregnenolone. This is the committed step. 3. Enzymatic modification. Pregnenolone is then shuttled between the mitochondria and smoother, undergoing various enzymatic modifications, e.g., hydroxylations, dehydrogenations, specific to the target hormone, e.g., cortisol, testosterone, estrogen. And 4. Secretion. Because they are lipid-soluble, steroid hormones are not stored but diffuse out of the cell into the bloodstream immediately after synthesis. 6. How does lipid-soluble hormones exert their effects intracellularly? Answer. Intracellular effects of lipid-soluble hormones. Lipid-soluble hormones, like steroids and thyroid hormones, can easily pass through the plasma membrane to exert their effects intracellularly. 1. Intracellular receptors. They bind to specific receptor proteins located in the cytoplasm or nucleus of target cells. 2. Hormone receptor complex. This binding forms a hormone receptor complex that moves into the nucleus, if binding occurred in the cytoplasm. 3. Gene expression regulation. The complex binds to specific DNA sequences, hormone response elements or HREs, near target genes. And 4. Protein synthesis. This binding acts as a transcription factor, either activating or inhibiting gene transcription, ultimately altering the synthesis of specific proteins that produce the cellular response. 7. Give an account of hormonal regulation of reproduction in seasonal breeders. Answer. Hormonal regulation of reproduction in seasonal breeders. Reproduction in seasonal breeders is timed to ensure offspring survival during optimal environmental conditions, primarily regulated by photoperiod, day length, input to the hormonal system. 1. Photoperiodism. Changes in day length are detected by the nervous system and relayed to the pineal gland, which secretes melatonin. 2. Melatonin's role. Melatonin acts on the hypothalamus to modulate the release of GnRH. 3. GnRH, FSH, LH. GnRH then regulates the pituitary's release of FSH and LH, which control gonadal activity, testosterone, estrogen, progesterone production. And 4. Timing reproduction. In long-day breeders, e.g., hamsters, decreasing melatonin in spring, summer stimulates the HPG axis. In short-day breeders, e.g., sheep, increasing melatonin in autumn, winter stimulates the axis. Translation. 1. MCQ. For this you can read guess papers. 2. Short notes. And 3. Long question. AI responses may include mistakes. Answers might be inaccurate or misleading. Double check for accuracy. Kinesis. Answer. Kinesis. Kinesis is a non-directional, or undirected, movement response of a motile organism or cell to a stimulus. The rate of movement or the frequency of turning depends on the intensity of the stimulus, but not its direction. The organism moves randomly until it reaches an area with a more favorable stimulus level, at which point its rate of movement decreases. B. Flocking in birds. Answer. Flocking in birds. Flocking in birds is a common social behavior where groups of birds, known as flocks, gather and move together. This behavior offers several survival advantages. 1. Predator defense. Large flocks can confuse predators, making it harder to target a single individual. 2. Foraging efficiency. Birds in a flock can more effectively locate food sources and share information about food availability. 3. Aerodynamic efficiency. Flying in formation can reduce energy expenditure during long migrations. And 4. Social interaction. Flocking facilitates mating opportunities and social bonding. C. Kin selection. Answer. Kin selection. Kin selection is an evolutionary strategy that favors the reproductive success of an organism's relatives, even at a cost to the organism's own survival or reproduction. This behavior is explained by inclusive fitness theory, which suggests that an individual's fitness is not just measured by its own offspring, but also by the number of shared genes passed on by its relatives. Altruistic behaviors, such as a worker bee sacrificing itself for the colony, are often a result of kin selection. D. Parental investment. Answer. Parental investment. Parental investment refers to the time, energy, and resources that a parent provides to their offspring to increase the offspring's chances of survival and reproductive success, at the cost of the parent's ability to invest in other offspring. Examples vary widely across species, from birds building nests and feeding chicks to mammals providing milk and protection. The level of investment often differs between sexes, driven by evolutionary pressures. E. Habituation. Answer. Habituation. Habituation is a simple form of non-associative learning where an organism decreases or ceases its response to a stimulus after repeated or prolonged presentations. It is a form of learning that allows an organism to ignore irrelevant stimuli and focus on important or novel ones. For example, a bird may initially fly away when a car passes, but over time, it becomes habituated to the noise and stops reacting. 3. What is learned behavior? Discuss trial and error learning, reasoning and cognition behavior. Answer. Learned behavior. Learned behavior is behavior that is modified as a result of experience. This contrasts with innate, or instinctive, behaviors which are genetically programmed. Learned behaviors allow organisms to adapt to changing environments. 1. Trial and error learning. This involves an organism attempting various responses to a problem until it finds one that works. Unsuccessful behaviors are gradually eliminated, and successful ones are reinforced, leading to the learning of the correct response. 2. Reasoning. This is a more complex form of learning that involves problem solving through insight and mental manipulation of information, without direct physical trial and error. It often involves applying past experiences to new situations to find a solution. And 3. Cognition behavior. This encompasses a range of mental processes including thinking, knowing, remembering, and problem solving. Cognitive behavior is often associated with higher intelligence and complex nervous systems, allowing for sophisticated learning and decision making. 4. Give an account of social organization of primates. Add a note on its significance. Answer. Social organization of primates. Primates exhibit a wide variety of complex social structures, ranging from solitary individuals to large, multi-male, multi-female groups. Common organizations include 1. Solitary. Individuals live alone but interact for mating, e.g., orangutans. 2. Monogamous pairs. A male and female live together with their offspring, e.g., gibbons. 3. Uni-male groups. One male lives with multiple females and their young, e.g., gorillas. And 4. Multi-male, multi-female groups. Multiple males and females live together in a complex hierarchy, e.g., chimpanzees, baboons. The significance of these social structures includes enhanced protection from predators, improved foraging efficiency, and opportunities for social learning and cooperation, which are crucial for survival and reproduction. 5. How fishes care for their young ones? Explain with suitable examples. Answer. Parental care in fishes. Parental care in fish is diverse and can include guarding eggs, building nests, carrying eggs or young in the mouth brooding, or even providing nutrients directly. 1. Nest building and guarding. Male sticklebacks build nests and guard the eggs until they hatch. 2. Mouth brooding. Cichlids are a well-known example where parents carry eggs and fry in their mouths for protection. 3. Internal fertilization and live birth. Some fish, like guppies, give birth to live, fully developed young, providing protection during the most vulnerable developmental stages. And 4. Nutrient provision. Discus fish secrete a mucus from their skin that their fry feed on. 6. Discuss different aspects of aggressive behavior. Answer. Aggressive behavior. Aggressive behavior in animals encompasses a range of actions intended to harm or intimidate another individual. It serves various evolutionary functions. 1. Resource defense, protecting food, territory, or mates. 2. Establishing dominance hierarchies, determining social rank within a group, which reduces future conflicts. 3. Sexual selection, males often fight for access to females. And 4. Parental defense, protecting offspring from predators or intruders. Aggression can range from displays of dominance, e.g., posturing, vocalizations, to physical conflict, with many species using ritualized fighting to minimize serious injury. 7. Write an essay on migration in birds. Answer: Migration in birds. Bird migration is the regular, seasonal movement, often north and south, undertaken by numerous species. This behavior is driven by the need for better food resources and breeding grounds. 1. Triggers: changes in day length, photoperiod, and temperature are key environmental triggers. 2. Navigation: birds use a variety of cues for navigation, including the sun, stars, the Earth's magnetic field, and visual landmarks. 3. Physical demands: migration requires significant energy reserves, often built up through hyperphagia, excessive eating, before the journey. 4. Routes: migratory routes, or flyways, are often consistent from year to year and can span thousands of kilometers across continents and oceans. And 5. Significance: migration is essential for species survival, allowing them to exploit seasonal abundance of food and avoid harsh weather conditions. AI responses may include mistakes. Answers might be inaccurate or misleading. Double check for accuracy. 3. पर्यावरणीय नीतिशास्त्र से आप क्या समझते हैं? विवेचना करें. आंसर: पर्यावरणीय नीतिशास्त्र व्यवहारिक दर्शन की एक शाखा है जो पर्यावरण के संरक्षण से संबंधित नैतिक समस्याओं का अध्ययन करती है, यानी मनुष्य और पर्यावरण के संबंधों का नैतिक सिद्धांतों के आलोक में अध्ययन करती है. है. 2. अमेरिकी विद्वान एल्डो लियोपोल्ड ने भूमि नैतिकता, लैंड एथिक्स, की अवधारणा प्रस्तुत की, जिसमें कहा गया कि भूमि को केवल संसाधन मात्र नहीं समझना चाहिए, बल्कि एक ऊर्जा प्रवाह के रूप में देखना चाहिए जिसमें जीव जंतु और पौधे पनपते हैं. 3. पर्यावरणीय नैतिकता के दृष्टिकोण से, वनोन्मूलन जैसी समस्याएं कई नैतिक चिंताओं को जन्म देती हैं, जैसे जैव विविधता का ह्रास, जलवायु परिवर्तन, और स्वदेशी समुदायों का विस्थापन. एंड 4. ये पर्यावरणीय मूल्यों की वैचारिक नींव के साथ साथ जैव विविधता एवं पारिस्थितिकी प्रणालियों की रक्षा एवं रखरखाव के क्रम में सामाजिक दृष्टिकोण, कार्यों तथा नीतियों से संबंधित मुद्दों का अध्ययन करती है. 4. पीपल फ्रेंडली तथा इको फ्रेंडली उत्पादन से आप क्या समझते हैं? विवेचना करें. आंसर: पीपल फ्रेंडली उत्पादन का अर्थ है ऐसी उत्पादन प्रक्रियाएं जो मानव कल्याण और अधिकारों को प्राथमिकता देती हैं, जैसे सुरक्षित कामकाजी परिस्थितियां और उचित वेतन, जबकि इको फ्रेंडली, पर्यावरण के अनुकूल, उत्पादन का अर्थ है ऐसी प्रक्रियाएं जो पर्यावरण पर मानवीय गतिविधियों के हानिकारक प्रभावों को रोकने या कम करने के लिए तैयार की गई हैं. 1. इको फ्रेंडली उत्पादन में प्राकृतिक संसाधनों के दुरुपयोग को रोककर संतुलन स्थापित करने की आवश्यकता होती है. 2. इसका उद्देश्य आर्थिक विकास और पर्यावरणीय संरक्षण के बीच संतुलन स्थापित करना है. 3. इन उत्पादन प्रक्रियाओं में पर्यावरण संरक्षण की पारंपरिक गतिविधियों को व्यवहार में लाने की आवश्यकता होती है. एंड 4. ये टिकाऊ उत्पादन, सस्टेनेबल प्रोडक्शन, के सिद्धांतों से निकटता से जुड़ा है, जो भविष्य की पीढ़ियों के लिए संसाधनों को संरक्षित करने पर भी ध्यान केंद्रित करता है. 5. संस्कृति आधारित जेंडर विभेदीकरण से आप क्या समझते हैं? विवेचना करें. आंसर: संस्कृति आधारित जेंडर विभेदीकरण का अर्थ है वे सामाजिक और सांस्कृतिक प्रथाएं, मान्यताएं और मानदंड जो व्यक्तियों के बीच उनके लिंग के आधार पर असमान व्यवहार या भेदभाव को बढ़ावा देते हैं. 1. यह अवधारणा समाज द्वारा निर्धारित लिंग भूमिकाओं, अपेक्षाओं और पदानुक्रमों से उत्पन्न होती है, न कि जैविक अंतरों से. 2. इसमें अक्सर महिलाओं या पुरुषों के लिए कुछ व्यवसायों, शिक्षा के अवसरों या सामाजिक भूमिकाओं को प्रतिबंधित करना शामिल होता है। 3. ये पितृसत्तात्मक संरचनाओं और पारंपरिक मान्यताओं के माध्यम से कायम रह सकता है जो एक लिंग को दूसरे से श्रेष्ठ मानते हैं। एंड 4. इस तरह का भेदभाव अक्सर उन समुदायों में गहराई से निहित होता है जहां पारंपरिक मूल्य आधुनिक समानता के विचारों पर हावी होते हैं। 6. व्यावसायिक नैतिकता के आलोक में विवेकानंद के वेदांतिक नैतिकता के महत्व की व्याख्या करें। आंसर: विवेकानंद की वेदांतिक नैतिकता, जो सभी प्राणियों में एक ही दिव्य चेतना, ब्रह्म, का सिद्धांत सिखाती है, व्यावसायिक नैतिकता के आलोक में अत्यंत महत्वपूर्ण है क्योंकि यह स्वार्थ से परे जाकर सेवा, ईमानदारी और सार्वभौमिक भाईचारे को बढ़ावा देती है। 1. वेदांतिक नैतिकता के अनुसार व्यवसाय का उद्देश्य केवल लाभ कमाना नहीं, बल्कि नर सेवा ही नारायण सेवा, मानव सेवा ही ईश्वर सेवा है, के सिद्धांत का पालन करना होना चाहिए। 2. ये कर्मचारियों, ग्राहकों और समाज के प्रति ईमानदारी और नैतिक जिम्मेदारी पर बल देती है, क्योंकि सभी में एक ही ब्रह्म का वास है। 3. यह व्यावसायिक नेताओं को करुणा, निस्वार्थता और सामाजिक कल्याण की भावना के साथ कार्य करने के लिए प्रेरित करती है, जिससे एक न्यायपूर्ण और टिकाऊ व्यावसायिक वातावरण बनता है। एंड 4. यह आधुनिक कॉर्पोरेट सामाजिक जिम्मेदारी (CSR) की अवधारणा के साथ संरेखित होती है, जिसमें व्यवसाय केवल शेयरधारकों के लिए नहीं बल्कि सभी हितधारकों के लिए जवाबदेह होते हैं। AI responses may include mistakes. Answers might be inaccurate or misleading. Double check for accuracy. 2. निम्नलिखित में से किन्हीं 4 पर संक्षिप्त टिप्पणियां लिखें। यह एक वैचारिक गृहकार्य समस्या है जिसमें किसी विशिष्ट विषय पर संक्षिप्त नोट लिखने की आवश्यकता होती है। चूंकि छवि में कोई विशिष्ट प्रश्न नहीं पूछा गया है, इसलिए प्रश्न का उत्तर नहीं दिया जा सकता है। कॉर्पोरेट नैतिकता, कॉर्पोरेट मोरालिटी। आंसर: कॉर्पोरेट नैतिकता। कॉर्पोरेट नैतिकता से तात्पर्य उन नैतिक सिद्धांतों और मूल्यों से है जो किसी संगठन के भीतर निर्णय लेने और व्यवहार को निर्देशित करते हैं। इसमें ईमानदारी, निष्पक्षता, जिम्मेदारी और पारदर्शिता जैसे गुण शामिल हैं। ये सुनिश्चित करता है कि व्यवसाय कानूनी और नैतिक रूप से संचालित हों, हितधारकों के हितों की रक्षा करें और समाज में सकारात्मक योगदान दें। B. समग्र विकास, होलिस्टिक डेवलपमेंट। आंसर: समग्र विकास। समग्र विकास एक दृष्टिकोण है जो किसी व्यक्ति के शारीरिक, भावनात्मक, मानसिक, सामाजिक और आध्यात्मिक कल्याण के विकास पर ध्यान केंद्रित करता है। ये शिक्षा और विकास के लिए एक संतुलित और व्यापक दृष्टिकोण है, जिसका उद्देश्य व्यक्तियों को जीवन के सभी पहलुओं में अपनी पूरी क्षमता हासिल करने में मदद करना है। C. कार्य संस्कृति एवं नैतिकता, वर्क कल्चर एंड एथिक्स। आंसर: कार्य संस्कृति एवं नैतिकता। कार्य संस्कृति किसी संगठन के भीतर साझा किए गए मूल्यों, मान्यताओं और व्यवहारों का समूह है, जबकि कार्य नैतिकता कर्मचारियों के नैतिक सिद्धांतों और मानकों को संदर्भित करती है। एक मजबूत कार्य संस्कृति और नैतिकता उत्पादकता, कर्मचारी संतुष्टि और सफलता को बढ़ावा देती है। D. जेंडर का समाजशास्त्रीय पक्ष, सोशियोलॉजिकल एस्पेक्ट ऑफ जेंडर। आंसर: जेंडर का समाजशास्त्रीय पक्ष। जेंडर का समाजशास्त्रीय पक्ष उन सामाजिक रूप से निर्मित भूमिकाओं, व्यवहारों, अभिव्यक्तियों और पहचानों से संबंधित है जो समाज पुरुषों और महिलाओं के लिए उपयुक्त मानता है। ये जैविक लिंग से अलग है और इस बात की पड़ताल करता है कि समाज लिंग मानदंडों को कैसे आकार देता है और व्यक्तियों के जीवन को कैसे प्रभावित करता है। E. जेंडर का मनोवैज्ञानिक पक्ष, साइकोलॉजिकल एस्पेक्ट ऑफ जेंडर। आंसर: जेंडर का मनोवैज्ञानिक पक्ष। जेंडर का मनोवैज्ञानिक पक्ष लिंग पहचान, लिंग भूमिका, व्यवहार और लिंग टाइपिंग जैसे संज्ञानात्मक और भावनात्मक पहलुओं की पड़ताल करता है। ये जांच करता है कि व्यक्ति अपनी लिंग पहचान कैसे बनाते हैं और ये मनोवैज्ञानिक कारक उनके विचारों, भावनाओं और व्यवहारों को कैसे प्रभावित करते हैं। F. परिवार के विस्तार के रूप में समाज, सोसाइटी इज एन एक्सटेंशन ऑफ फैमिली। आंसर: परिवार के विस्तार के रूप में समाज। यह अवधारणा बताती है कि समाज परिवार की एक व्यापक इकाई है, जहां सामाजिक संरचनाएं, मूल्य और रिश्ते पारिवारिक गतिशीलता से उत्पन्न होते हैं। ये परिवारों और बड़े समुदाय के बीच परस्पर संबंध और एक दूसरे पर निर्भरता पर जोर देता है। G. नियोक्ता के कर्तव्य, ड्यूटीज ऑफ एम्प्लॉयर्स। आंसर: नियोक्ता के कर्तव्य। नियोक्ता के कर्तव्यों में कर्मचारियों को एक सुरक्षित और स्वस्थ कार्य वातावरण प्रदान करना, उचित वेतन और लाभ सुनिश्चित करना, गैर भेदभावपूर्ण प्रथाओं को बढ़ावा देना और सभी लागू श्रम कानूनों और विनियमों का पालन करना शामिल है। AI responses may include mistakes. Answers might be inaccurate or misleading. Double check for accuracy. A. Principles of ethics. Answer. Pillars of right and wrong. Ethics are well-established standards that determine what humans should do, typically in terms of rights, obligations, societal benefits, fairness, or specific virtues. These standards are often based on four core principles: beneficence, doing good; non-maleficence, doing no harm; autonomy, respecting self-determination; and justice, fairness. B. Integrity in work ethics. Answer. Adherence to moral principles in the workplace. Integrity is evidence of ethical behavior within organizational processes, adhering to standards of truthfulness, transparency, and honesty. Work ethics provide rules for how individuals should behave toward others and institutions in a professional setting. C. Courage and sympathy is professional ethics. Answer. Virtues demonstrating professional character. Virtue ethics views the manifestation of qualities like courage and compassion, sympathy, as core principles of morality. These virtues are among the 12 common ethical principles in professional settings, which also include honesty, accountability, and respect. Translation. 3. What is ecologically responsible technology? Answer. Ecologically responsible technology, or environmentally responsible technology, involves the design and implementation of technology that maintains a balance between human needs and environmental protection. It ensures that development projects contribute to a healthy planet by minimizing harm to ecosystems and promoting sustainability. This field relates to ecology, which is the study of how organisms interact with their environment and each other. The goal is to better manage natural resources, mitigate pollution impacts, and protect human health and endangered species. 4. What is professional ethics? Answer. Professional ethics is a branch of moral philosophy that defines the principles and standards of conduct governing a specific profession or field. It involves understanding the moral obligations of individuals within their professional roles. These ethics ensure that professionals act responsibly and with integrity, considering the impact of their actions on clients, colleagues, and society as a whole. It is a guiding force that ensures every person within a community takes care of their surrounding environment and community members. 5. Discuss the gender-based division of labor at present in India. Answer. Gender-based division of labor in India refers to the societal pattern where specific tasks and roles are assigned to individuals based on their gender, often resulting in men primarily engaging in paid, public sphere work and women primarily engaging in unpaid, domestic sphere work. 1. Persistence of traditional roles. Despite modernization and increased female education, traditional gender roles remain deeply embedded in Indian society. 2. Workforce participation disparity. There is a significant disparity in labor force participation rates between men and women, with women being largely represented in the informal sector or unpaid domestic work. 3. Occupational segregation. Women often dominate certain professions, e.g., teaching, nursing, while being underrepresented in others, e.g., STEM, leadership roles. And 4. Impact of social norms. Societal expectations and patriarchal norms heavily influence career choices and work-life balance challenges for women. 6. Explain the importance of Vedantic ethics of Sri Aurobindo. Answer. The Vedantic ethics of Sri Aurobindo emphasizes a holistic approach to life, integrating spiritual realization with action in the world. 1. Integral yoga. His philosophy advocates for integral yoga, which seeks not just individual liberation but the transformation of earthly life through the descent of the divine into material existence. 2. Evolutionary ethics. Aurobindo viewed ethics as part of a spiritual evolution, where human morality is a stage leading to a higher, supramental consciousness that transcends conventional good and evil. 3. Importance of inner growth. The focus is on inner spiritual growth and the realization of the divine within oneself and all beings, which naturally leads to ethical action. And 4. Universal brotherhood. His ethics promote the idea of universal brotherhood and the unity of all existence, moving beyond narrow self-interest to a collective, divine life. AI